The Human In Vitro Drug Transporter Dataset in the DIDB contains results from in vitro transporter studies where a drug is tested as an inhibitor or a substrate for a given human drug transporter and its variants. By typing a citation number in the search area, you will retrieve all collected data in DIDB documents containing the citation. By clicking on this citation, you will go to the data tree view. Study results are organized according to the overall effect and mechanism of the interaction. Using a single click on a plus sign, you will expand the next layer of data presentation, while double-clicking will display the full entry. For example, in this transporter substrate entry, dabrafenib, a kinase inhibitor for cancer treatment as an object, is a substrate of BCRP in MDCK transfected cells. While in this transporter inhibition entry, dabrafenib as a precipitant is an inhibitor of OATP1B1-mediated estradiol 17-beta glucuronide transport in CHO transfected cells. Transport parameters such as efflux ratio and IC50, along with detailed experimental conditions, are all curated. In vitro to in vivo prediction ratios using the basic model recommended in the FDA's DDI guidance are also presented when relevant I values are available. From this citation, you know that dabrafenib is an inhibitor of OATP1B1. If you are interested in all compounds that inhibit OATP1B1 with IC50 values less than or equal to 10, you can do the following query. From this citation page, click on Queries, then Transporter. This will take you to the Transporter query page. Click on Transporter with Precipitants, find precipitants which inhibit OATP1B1 with IC50 values less than or equal to 10 micromolar, then click Submit. By conducting this query, you will obtain a complete list of potent in vitro inhibitors of OATP1B1, which includes 771 entries at this time. The results are presented by default in a table. You also have an option to view the results presented in a tree. Here you can customize the data for further meta-analysis. You can select or deselect columns, and copy, download, or print the results. You can filter the results to a specific substrate, such as the OATP1B1 in vitro marker substrate, estradiol 17-beta glucuronide, or you can choose from the drop-down list. You can also perform an advanced table search where you can filter by one or several conditions. Additionally, you can reorder columns such as IC50 values and rearrange columns. Of note, by clicking on a citation number, you can go to the citation details of this entry together with all the other data entered under the citation, view the PK profile of the citation's drugs, or go to the citation in PubMed or Semantic Scholar. In DIDB, transporter queries allow you to retrieve an in vitro transporter dataset. In addition, you can use drug queries, find all studies, to find all curated data related to this drug, which includes in vitro transporter data. I invite you to check out our other video tutorials, which provide in-depth looks into the contents and functionalities of the DIDB. And that concludes this video tutorial on the Human In Vitro Drug Transporter Dataset. If you have any questions, just go to our website, druginteractionsolutions.org, and click on Contact Us.